This video will cover subtopic 3.5, Genetic Modification and Biotechnology. This is the first video of two. And so in this video, we will cover understandings related to biotechnologies. And then in the second video, we'll cover understandings related to that of cloning for animals and plants. So applications and skills that we will go through related to genetic technologies, as well as some guidance. So we'll start with the first biotechnology, which is gel electrophoresis. And gel electrophoresis is a process that is used to separate proteins or fragments of DNA according to size. So it will involve the separation of charged molecules such as DNA or RNA or proteins. And remember, DNA and RNA have an overall negative charge associated with them. Proteins will be provided or put in the solution such that they will be given an overall negative charge. And this charge is important because these molecules will be subjected to an electric field. So a current will be applied. And that charge will be will cause them to migrate and separate according to size. So samples of DNA, RNA, or proteins are placed into a material called a gel, which is basically a material that has a lot of pores in it. And at the top of these gels are like these little depressions called wells. Now, the samples of DNA, RNA, or protein are placed into these wells, and then the gel is immersed in a conducting fluid, basically what's called a buffer. That conducting fluid is what will allow the electric field to be applied. So you have a power source, basically like a big battery, that will apply an electric current. And basically you'll get two ends set up on the gel. So one end will be a cathode, one end will be an anode. You'll create a negative charge at that anode. So you'll have all these electrons flowing to the anode, creating that negative repulsion that will cause these fragments of DNA, RNA, or proteins to move away because they're going to have an overall negative charge, so they're going to be repulsed by that negative charge, and they will move through that gel, which is porous in nature. Now, the fluids that the DNA, RNA, and proteins are put into are going to have the same amount of charge per mass. Now, because of this, the gel electrophoresis of DNA fragments will separate them based on size only. So the same amount of charge per mass means that they will be repulsed equally by that cathode. And so the only thing that will determine how they migrate through the gel is their size. So proteins can be positively, remember those are histones, or negatively charged, so they can be separated according to their charge as well, but most proteins are going to be overall negatively charged. DNA, RNA, and proteins from eukaryotes are too long to move through the gel, so they have to be broken up into smaller fragments first. Smaller fragments move faster and are basically going to be closer to the positive electrode. So those smaller fragments are going to move quicker and end up at the anode faster, or end up closer to the anode faster. Interpretation of gel electrophoresis is done by comparing the unknown sample to what is called a DNA ladder loaded into one of the wells. A DNA ladder is a sample of known sequence and length. So, for example, in the picture shown here, one of these samples here would be the DNA ladder. The DNA ladder, we would know the size of each of these fragments, and so we can use the ladder to determine the size of the fragments in our sample. So before we can use gel electrophoresis for DNA molecules, for example, we have to subject our DNA samples to PCR. So PCR can be used to amplify those small amounts of DNA, which can then be separated by gel electrophoresis. So PCR, just a reminder, is that polymerase chain reaction. So we're using that DNA polymerase, that Taq polymerase, to basically create regions of interest or fragments of interest from our DNA. And PCR is going to be used to make large numbers of copies of DNA. Just a reminder that the PCR cycle repeats 25 to 35 times in a typical reaction, which will generally take about 2 to 4 hours to complete. Now, depending on the length of the DNA region being copied, this can take closer to 4 hours, and if it's shorter, it can be like closer to 2 hours. If the reaction is efficient, if it works well, the target region can go from just one or a few copies to billions of copies. So we did cover those steps for PCR back in subtopic 2.7, so you can check out page 155 to 116 of your textbook, or go back and rewatch that video involving subtopic 2.7. So a few other things just to recall about DNA is that PCR basically relies on that thermostable DNA polymerase, so the DNA sample is going to be heated and cooled repeatedly. And so you need a polymerase that is going to be stable at those high temperatures. So the Taq polymerase is that DNA polymerase obtained from an aquatic species of bacteria that lives by thermal vents. And basically that PCR is also going to require DNA primers that are designed specifically for the DNA region of interest. Now, in PCR, the reaction is repeatedly cycled through a series of temperature changes, which will allow many copies of the target region to be produced. Those series of temperature changes are what's going to separate the DNA strands and then allow the primer to bind and allow the Taq polymerase to do its thing. So PCR has many research and practical applications. It is routinely used in DNA cloning, medical diagnostics, and forensic analysis of DNA. So a few applications of PCR is that the results of a PCR reaction are usually visualized. They're made visible by using gel electrophoresis. So once you've amplified your DNA or your various regions of DNA, you can view those created fragments using gel electrophoresis. So PCR is used to amplify genes associated with genetic disorders from the DNA of patients or from fetal DNA. This is part of the test for genetic diseases. And PCR can also be used to test for bacterium or DNA virus in a patient's body. This is the test that's currently being used, or the method that's being currently used to scan people or to test people for the presence of coronavirus. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or tissue sample. So right now, cheek swabs or mouth swabs are being collected, and then that DNA is being subjected to PCR to determine if that virus is present or not. It can be used to test for the presence of genetically modified food ingredients as well by using a primer that binds to a genetically modified DNA sequence. So another biotechnology that's commonly used is one of gene transfer. And this is the transfer of genes from one species to another. And when you do this, this is known as genetic modification. Remember that genetic modification is possible since the genetic code is universal. So those same codons that are found in RNA, are used to code for amino acids, are the same for all species that are classified as living. Genes, therefore, can be transferred from eukaryotes to bacteria. So an early example of this transfer was the gene for making human insulin was transferred to bacteria. And this was done so large quantities of insulin could be made easily to treat diabetes by taking that gene that codes for human insulin, putting it into bacteria. The bacteria would now make large quantities of that insulin, which could then be purified and given to people who have diabetes. The technique of gene transfer has also been used to introduce new characteristics to animal species. So, for example, the goats that produce spider milk and genetically modified crops, especially. So, there are some techniques for transferring DNA into bacteria. And before we get into talking about that, just a few definitions that you need to know, and a few that you need to recall. So, when transferring genes into bacteria, you need to use plasmids. And just recall that a plasmid is a small ring of bacterial DNA that is separate from chromosomal DNA, which can replicate independently. Plasmids are often exchanged between bacterial cells. To be able to insert that DNA into those plasmids, you need to use what are called restriction enzymes, which are also known as restriction endonucleases. These are enzymes that will cut a DNA molecule at a particular place. So these enzymes will basically cut that plasmid open and allow a gene to be inserted that way. Now, once that plasmid has been cut open, you need to close it back up with, another enzyme called DNA ligase. And this is an enzyme that will join DNA molecules together by forming the sugar phosphate bonds between nucleotides. And it's used to seal nicks formed by what are called sticky ends, and we'll cover those in a second. So once you've inserted your DNA of interest into the plasmid, you need to close things back up, and you use a DNA ligase for that. Now, there can be two types of ends generated by restriction enzymes. You can get what are called sticky ends, and you can get blunt ends. So sticky ends are a fragment of DNA which are often produced by a staggered cut on the DNA using restriction enzymes, in which unpaired nucleotides of the strands are kind of unpaired or left hanging, if you will. So this picture down here demonstrates a sticky end. So here is your DNA sequence, and then this little gray area here that I'm just outlining in purple represents where the endonuclease, or where that restriction enzyme, would cut. And so when it cuts the DNA, it's cutting it here, but then it's kind of pulling it apart and then making that last cut a little further down the DNA. So now what you end up with is, here you have paired nucleotide, and then here you have a series of unpaired nucleotides. So they're just hanging out there. If you have a gene of interest that has a complementary sequence of unpaired nucleotides, then it can easily attach to this unpaired sequence. So they'll basically want to stick together. Now, you can also generate what are called blunt ends from restriction enzymes. Now, this is the end of a DNA fragment resulting from the breaking of a DNA molecule in which there are no unpaired bases. Hence, both strands are of the same length, basically. So here is the DNA sequence. Here is where the restriction enzyme is going to cut. It basically cuts it right between the nucleotides, so that you end up with these ends that have paired nucleotides. There are no unpaired nucleotides here. It can be a bit harder to get DNA fragments inserted in between blunt ends, so you often aim for the generation of sticky ends. So now, taking all this into account, you can work through the steps for gene transfer. So in this example here, we'll look at a gene, the gene for insulin, being inserted into a plasmid, and then that plasmid inserted into a bacterial cell. That bacterial cell then now grown and is used to extract insulin, or obtain insulin from. So in this case, you need the mRNA sequence, and this gets extracted from the pancreatic cells, and that mRNA is then treated with an enzyme called a reverse transcriptase. So a reverse transcriptase is an enzyme that will take RNA and will convert it into a complementary DNA. Um, we didn't talk about it in the course, but you can't necessarily take the DNA sequence encoding for the gene because there can be modifications to the RNA sequence after it has been transcribed. So you want to take the mRNA sequence that would be used for transcription. So you take that mRNA sequence and then you can convert that into a complementary DNA strand. Then you can take your plasmid, which is obtained from a bacterial cell, and cut it with restriction enzymes. And you'll cut it with restriction enzymes that will generate those sticky ends. And those sticky ends are shown here. I've highlighted that in purple, so you get those unpaired nucleotides. And you also want to treat your gene with the same restriction enzyme to generate the same type of sticky ends. This will help the gene insert into the plasmid. So so in this case, the insulin gene is going to be cut with the same restriction enzyme as the plasmid, and both will have sticky ends. So plasmid will have sticky ends, insulin gene will have sticky ends. The gene and the plasmid are then spliced together by the enzyme DNA ligase to form a recombinant product. So we take our insulin gene, we insert it into the plasmid using DNA ligase. Here's our insulin gene now inserted into the plasmid. And then that recombinant plasmid that contains that insulin gene is then introduced into the host cell. So we put this back into the bacteria cell, and then the bacteria are placed into culture broth and grown. And as they grow, they should be producing that insulin RNA and making protein from that. So they'll start to create insulin protein. And then the insulin can be separated and purified and can now be used by diabetic patients. And this process has revolutionized how diabetes is treated. So with genetically modified crops now, the same process of introducing a gene can be used to create genetically modified crops. And so there's a lot of pros and cons to genetically modified crops. There's a lot of benefits to them. So in most cases, you can improve the nutritional value of food by allowing this food to produce proteins or vitamins or sneak in some vaccines in them. The crops can be produced that lack known allergens, so you can remove genes that may cause allergies in people. Crops can grow in arid conditions for better yield, so you can introduce drought-resistant genes into these plants. Genetically modified crops can produce herbicides to kill pests, and this is one of the examples that we'll look at. And they can be used to improve food supply as well. So the genetically modified crops can also have longer shelf lives, and they can help reduce economic costs and carbon footprints as well. Now, these are all the good things about, or all the benefits of genetically modified crops. Now, there are some potential risks of using genetically modified organisms as well. So the new traits that are introduced could cause severe health reactions. So if you are putting in a new protein or putting in a new gene, this may cause an unintended allergic response in certain portions of the populations. Removal of traits could also have unknown effects. So, we may understand how one protein might affect a particular trait, but by removing that protein, it may affect how other proteins get expressed. And by now introducing these genetically modified crops, we're basically going to reduce biodiversity in these local environments. And, if you get cross-pollination of these genetically modified crops with plants that are considered weeds, you could lead to the creation of superweeds potentially. Now, because these genetically modified organisms are created in a lab, they often have patents associated with them, and these patents come with severe restrictions on how farmers can use these seeds. The other thing that a lot of people are working towards or trying to get done is that food with genetically modified components should be labeled. So you may be consuming food that may not have a label indicating that it contains genetically modified organisms. And different governments may have conflicting regulatory standards concerning safe usage. So in Europe, for example, there is very strict limitations on the use of genetically modified crops versus here in North America. So one of the things that you'll be expected to do or could be expected to do is analyze the data on risks to monarch butterflies to a genetically modified type of corn referred to as Bt corn. Now, Bt corn is a genetically modified maize that incorporates an insecticide produced from a particular bacteria. Now, this insecticide is lethal to certain types of larvae, particularly the European corn borer, which would otherwise eat the crop. So the European corn borer can devastate crops of corn pretty easily. So these genetically modified corn plants produce a natural pesticide that will help reduce damage from this particular pest. Now, concerns have been raised that the spread of Bt corn may also have an impact on the survival rates of monarch butterflies. So while monarch butterfly larvae feed exclusively on milkweed, wind-borne pollen from Bt corn may end up on nearby milkweed plants. So that pollen from these corn plants may inadvertently end up on the leaves of the plants that monarch butterfly would eat. And so they might inadvertently eat that pollen containing that pesticide, and that may have a potential effect on population levels of monarch butterflies. In 1999, a preliminary study was conducted investigating the association between the exposure of Bt corn pollen and survivor rates among monarch caterpillars. Now, monarch caterpillars were fed milkweed leaves that had been dusted with pollen from Bt corn. This was done to simulate spread through wind. Growth and mortality rates were compared against caterpillars fed non-dusted leaves or leaves dusted with non-genetically modified pollen. Caterpillars exposed to Bt pollen were found to have eaten less, grew more slowly, and exhibited higher mortality rates. So here are the results obtained from that study. The red are the milkweed plants dusted with Bt pollen. And the survival rate here, you can see significantly drops of those monarch caterpillars that had eaten from these particular plants. Now, some scientists suggested that the results may lack validity because they do not accurately reflect natural conditions. Now, some of the issues with this study was that there were higher levels of Bt pollen on the leaves than would be found naturally. So they didn't take into account that rain might diminish buildup of this pollen on the leaves. And larvae were restricted in their diet, whereas in the field, larvae could potentially avoid eating pollen-dusted leaves. So to address some of these issues, another study was done, which it compared survivor rates of monarch butterflies based on proximity to Bt cornfields. What was found was that there was no significant increase in mortality when monarch larvae were placed in or near an actual Bt cornfield. From this, it was concluded that exposure to Bt pollen poses no significant risk to monarch butterfly populations. Some of the data from the study is shown here. So here we have time of days, basically from when Bt larvae were placed in their area. So the blue squares represent larvae that were placed near cornfields that contain that genetically modified corn. The red triangles represent the larvae placed near normal fields. And as time went on, you can see that the survival rates between the two larvae really aren't that different or significantly different. So this is the end of the video for part 1 of 3.5.